This lecture series titled Data Visualization in Bioinformatics and Systems Biology is organized by 2 universities, and covers subjects ranging from general biology to medicine. However, a common thread across all topics is the goal of data visualization. In this lecture, after a short introduction, I will briefly summarize some medical topics of the course. Bioinformatics is the application of computer technology to the understanding and effective use of biological and biomedical data. Bioinformatics is a unique scientific field which includes databases, methods and applications, programs, computational infrastructure, and data visualization. There are some topics definitely not included in bioinformatics. For example, statistical computations can be performed using a paper and a pen, thus there is no need for a computer. Similarly, simple equations, regardless of subject, are generally not included. Finally, programming by itself belongs to mathematics, and bioinformatics only uses programming as a tool to solve a specific problem. In this course, data visualization, a subfield of bioinformatics, will be covered. Bioinformatics has become an independent scientific field. This was also acknowledged by the Hungarian Academy of Sciences last year, when the Interdepartmental Scientific Committee on Bioinformatics was established. This means that scientific matters related to bioinformatics will not be discussed within the fields of medicine, biology, chemistry, or mathematics anymore, because bioinformatics has become an independent discipline. It is very important that this committee was established, because now not only is it possible to acquire a PhD degree in bioinformatics, but also one can become a doctor of the academy. Of note, across all scientific fields there are only 5 interdepartmental scientific committees of the Hungarian Academy of Sciences, and therefore we are very proud to have this honor. Please allow me to briefly draw your attention to 2 organizations promoting bioinformatics in Hungary. The Hungarian Society for Bioinformatics was established in 2011. The aim of the society is to strengthen the bioinformatics research in Hungary by making a bridge between mathematicians, computer scientists, biologists and pharmacologists. The society has been organizing seminars, meetings, conferences and actively participates in bioinformatics education in Hungary. The yearly bioinformatics conferences organized by the society brings together all researchers who are active in this field. Elixir is an intergovernmental organization that brings together life science resources from across Europe. These resources include databases, software tools, training materials, cloud storage and supercomputers. The goal of Elixir is to coordinate these resources so that they form a single infrastructure. This infrastructure makes it easier for scientists to find and share data, exchange expertise, and agree on best practices. Elixir includes 22 members and 1 observer, bringing together over 220 research organizations. It was founded in December 2013 and the Hungarian node was established in 2017. I encourage everyone interested in bioinformatics or in research related to bioinformatics to check out these organizations. Our data visualization course is organized jointly by Semmelweis University, Department of Bioinformatics and Pázmány Péter Catholic University, Faculty of Information Technology and Bionics. This means that we will have lecturers from both universities. Accordingly, the topics of the course will be covered from both medical and biological perspectives. Practically, data visualization topics with primarily medical focus will be covered by lecturers from Semmelweis University, while topics with chemical and biological focus will be covered by lecturers from Pázmány University. The lectures presented in the Pázmány University are going to be summarized in a later introduction. But for now I will briefly summarize the topics of medical focus. Let's start with the first lecture. Among the medically relevant graphical representations, first we will discuss 2 widely used visualizing methods, the Kaplan-Meier plot and the box plots. Survival analysis is a statistical procedure for data analysis, in which the outcome variable of interest is the time until an event occurs. The Kaplan-Meier curve is an estimator used to estimate the survival function. The Kaplan-Meier curve is the visual representation of this function, that shows the probability of an event at a respective time interval. The curve should approach the true survival function for the population under investigation, provided the sample size is large enough. We will provide an in-depth description as well as detailed guideline for making a Kaplan-Meier plot. Box plots is basically an interval scale that is used to perform estimation, which necessarily performs the abstraction of the data. Box plots are used to interpret and analyze the data. It can also be used to visualize the data. Box plots is a graphical method, which is used to illustrate the variation of the data in the data distribution. A histogram can also be used to display the data. However, if we compare a boxplots and a histogram, the latter offers only a sufficient display, while a boxplots also provides additional information corresponding to the multiple sets of data, which is displayed in the same graph. To visualize logical correlations, we will provide a description for two tools. Ishikawa diagram, otherwise known as the fishbone diagram, is one of the tools for this purpose. The aim of this tool is to list all potential root causes leading to a problem. A flowchart is a picture of boxes that indicates the process flow in a sequential manner. Since a flowchart is a pictorial representation of a process or algorithm, it's easy to interpret and understand the process. For genomic data visualization, we will show you the IGV and Cytoscape softwares. IGV, or Integrative Genomics Viewer, is a high-performance, easy-to-use, interactive tool for the visual exploration of genomic data. It supports flexible integration of all the common types of genomic data and metadata, which is investigator-generated or publicly available, and can be loaded from local or cloud sources. Cytoscape is an open-source software platform for visualizing molecular interaction networks and biological pathways and integrating these networks with annotations, gene expression profiles, and other state data. Although Cytoscape was originally designed for biological research, now it is a general platform for complex network analysis and visualization. Cytoscape core distribution provides a basic set of features for data integration, analysis, and visualization. Additional applications are available for network and molecular profiling analyses, new layouts, additional file format support, scripting, and connection with databases. A third option for genomic data visualization is clustering. Hierarchical clustering, also known as hierarchical cluster analysis, is an algorithm that groups similar objects into groups called clusters. The endpoint is a set of clusters, where each cluster is distinct from every other cluster, and the objects within a cluster are broadly similar to each other. We also have to mention the swimmer plot, which is another clinically frequently used plot. A swimmer plot is a graphical way of showing multiple pieces of a subject's response in one glance. The graph includes a bar showing the length of treatment for each patient, classified by the stage of the disease at baseline. Each line represents one patient in the study. If you are a researcher, analyst, data geek, art director, illustrator, or visual artist, who is seeking to explore or communicate a data set, or to think outside the box, and inside a circle, Circos is worth looking into. Circos is a software package for visualizing data and information. Circos can be used to display any kind of information. It's particularly suitable for layering different data sets to create highly informative infographics with texture and visual appeal. It visualizes data in a circular layout. This makes Circos ideal for exploring relationships between objects or positions. Circos attempts to bring a different aesthetic to science and strike a balance between flexibility and ease of use. Circos makes no assumptions about your data, uses extremely simple input data format, and makes image creation and customization easy. It helps to make science look better, one figure at a time. In the last medical section, we will talk about monitoring the activity of large neuronal populations with high spatiotemporal resolution. In this lecture, we will provide an introduction to the state-of-the-art high channel count neural devices developed to record extracellular spiking activity of neurons with high spatial density. There will be a demonstration of basic data visualization, processing, and analysis methods used in this field. We will also give a detailed demonstration of a frequently used data processing method called spike sorting, and we will show a short tutorial on our currently used spike sorting workflow, using a high-density electrophysiological recording, obtained in the laboratory in the Research Center for Natural Sciences. We have prepared the lectures in a way that first you get the relevant theoretical background for each topic. Then, the tools and methods will be summarized, which are useful for this particular purpose. In the end, we will provide useful references for further reading. There are also some exercises for some of the lectures for you to practice. For more information in general about the discussed topics, I suggest the book entitled Bioinformatics and Functional Genomics, published by Jonathan Pevsner. The book is somewhat heavy with more than 1,000 pages, but it provides a comprehensive summary. We will provide more targeted references for more information after each lecture. Finally, I would like to thank the help of Victoria, who helped us to prepare and improve our slides, so that we can have the best possible English texts. We will ask you about your opinion regarding the data visualization course. We will be interested in which lectures you like the most and the least. We kindly ask all of you to fill out our questionnaire, because this will help us to improve our course in the next year. A web link to access the questionnaire will be provided to you at the end of the semester. Thank you for your attention.